Hey everybody, what's going on? It's Mr. Dan here, and today I'm going to give you guys a lesson about gene regulation. Okay, so I'm actually not going to use this today. We're going to go to something else. We're going to start here, you guys, talking about gene expression. This is this here is a simulator that. This is just a simulation program that's going to look at DNA transcription and translation and some kind of like what goes on with it. So I'll make sure to put the link here. This is using PET interactive simulations. So these things are really great. I encourage you to come and play with this. And I want to start by just kind of like showing you a little bit about how this works. I don't want to do it too much because I want you to play with it yourself. But let's go forward here. So there's three different modes you can do. It says there's expression, mRNA, and multiple genes. So I'll show you each of these, starting here with expression. And so starting off, let's look at everything that we have. So right down the middle, the obvious thing, we've got DNA, double-stranded DNA in its double helix form. And you can see we've got the two, it's got one red and one blue. The purpose here is just to help us track one side versus the other. So we've got that. We've got a gene highlighted here. This is gene 1. There's multiple genes, gene 2 and gene 3. Okay. And each of these genes is going to create a different type of protein. Okay. So we're going to try to make all three proteins. We got diamonds, rhombus, if you like it. Tilt your head, it becomes a square. We got a hexagon and we got a cylinder. Okay. So we're going to make all three of these proteins. Each protein requires a different gene. You know that, of course, right? Okay, and so within the gene, there's two areas. We have the transcribed region, here in orange for this one, and then there's the regulatory region. So we're going to have to figure out what that means, but we're talking about gene regulation, so that's going to be the focus for us. And then up here, we've got a toolbox of different stuff that we can use. So I want to copy a gene, right? We got to do transcription first, and so I know that if I want to do transcription, I need RNA polymerases. So I'm going to throw out these two RNA polymerases. I can grab two. Let's just do one, okay? And I need this to bind to the DNA. That's what's got to happen. So I'm going to go ahead and place this in the spot where it belongs. I'm going to put it here, and we see what happens. It looks like it fixes to it. And then it dissociates, and it doesn't come back. Right, let's give that a try again. Let's put it there closer. Again, it doesn't stick to it, okay? So what that means is that it needs a little bit of help to stick on there, okay? And so we're going to give it some help. We're going to dig into our toolbox, and if I look at these, I see this one here that says positive transcription factor, okay? So this is a chemical, probably it's a protein, okay? And the job of this protein is to enable or increase the polymerase's affinity for the DNA, right? So it's going to help it attach. So we're going to put this down here. And something this does also, along with helping the RNA polymerase to attach to the DNA, and then, as we see, open it up and copy the mRNA, is it's like a marker site, right? So this positive transcription factor, this protein, it's going to target a specific spot on the DNA where it's going to sort of like tell the polymerase, like, this is your starting point here, okay? So whenever that polymerase finds that positive transcription factor, it's going to know that's the spot for me to start transcribing. Polymerase can work anywhere, right? This molecule here, this polymerase, it can copy any gene on the entire genome, okay? So it needs to know where to go. So these positive transcription factors, they're a bit more specific in what they're in where they're targeting. So now that I've got some RNA, actually let's highlight how this works, right? I can put this on here. I can copy the gene two at a time. More if I had more polymerases, okay? I'm going to go ahead and make a bunch of these mRNAs. And now I'm going to throw out some ribosomes. I've only got 2 ribosomes, but what they're going to do is they're going to start attracting and attaching to those mRNAs, and they're going to start making proteins, okay? So the more RNA I put out there, the more proteins I can make. All right, so we'll let that kind of go for a while. Okay. So what's happening as these proteins get made, we can kind of help it out a bit. Put it nearby. As these proteins get made, we're going to start to accumulate a lot of them, and the cell needs to do something with that. So I'm going to take us over here to a cell model, and let's kind of take a closer look here a little bit. So here we're looking at an animal cell, and it's got some different parts to it. Here we've got the nucleus, okay, and this is where all the DNA is going to be held, and the nucleus, that's where this is happening, okay? The transcription is happening inside the nucleus, okay? And so then that RNA, it's going to leave the nucleus and it's going to head out into the cell, and it's going to start looking for something called ribosomes, okay? It's going to look for those where protein synthesis is going to happen. Those ribosomes are freely floating around the cell, and then they're also found attached to what we call the rough endoplasmic reticulum, which is an organelle that's right outside of the nucleus. It's a folded membrane, okay? So it's made out of a lot of lipids and stuff. Membrane just means it's a barrier. And so it's just this like folded sheet that's all around the nucleus, and it's got ribosomes all over it, okay? So this is going to do a couple of things. The ribosomes are going to make the proteins, and then because it's attached to this membrane, what's going to happen is that as the proteins start to accumulate in that area, they're going to get packaged up so that they can be delivered to another area. Okay, so what's that packaging going to look like? Well, they're going to come in these little, sorry, I'm trying not to circle it. These little circles here, okay, they're going to get packaged in those, like hundreds, thousands at a time maybe, are going to go into those little globules here, and they're going to be transported to what we call the Golgi body, okay, or sometimes the Golgi apparatus. Okay, and what's going to happen here is that the proteins, they're going to be sort of like maybe make some final adjustments to the shape of them, some subtle changes, and get them truly ready for whatever job they're supposed to have. And then they're going to get packaged up again and delivered to wherever they're supposed to go. And again, in these little things, we call them vesicles. Okay, so these little globules here, it's a vesicle. It just means it's a transport, like a little transport bubble. So from the Golgi, or from the endoplasmic reticulum to the Golgi body, where it's going to be sort of like processed, and then after that processing and final checks are done, it's going to go back into another vesicle where it might be delivered to somewhere in the cell, maybe to a mitochondria, maybe to these vacuoles, maybe to any parts we might find in here, or it may even go all the way out to the cell membrane, as a transport protein or some sort of like communications protein, whatever the job it might be. Okay, so it's going to be transported through those. So what that's going to look like here in our simulation is really we can just start grabbing these and we're going to place them up here. Okay, and so in doing this, we're modeling that action of putting them into a vesicle for transport to some other part in the cell. Okay, so I made 8 of these, and that's a pretty good number. So I don't think I need to make any more. Oh, maybe 9. Okay, I don't want to make too many more. I've got enough. And so once the body recognizes that it has enough of that particular protein, a signal is going to be sent that's going to tell this transcription factor that it no longer needs to attach. Okay, we'll go over how that's going to look in a little bit. So that's going to be gone. And then some other things might happen, like we might have some of these, like we call them mRNA destroyers, and these are enzymes that are going to come along and start to really like chomp up all that mRNA, and that's going to prevent proteins from being made any further. Okay, so showing us a little bit of this process here. So here we go. We got proteins being made on gene 1. Next gene we look at, it's got, it's the same thing. I want you guys to come and check it out. There's going to be other things that we could try to use I suppose here we could also put on a negative transcription factor, and this is going to prevent the RNA from binding as well. Okay, so I want you to go ahead and try out these different ones and see, just see how they work, right? Play around with these different transcription factors. Sometimes maybe you need multiples of them, see what happens when you put in the negative, try a different balance of polymerase versus ribosome versus mRNA, okay? And you can come and do that. Here, we can kind of play with some dials, like we can increase the concentration of polymerase. We can increase or decrease how strongly transcription factors are going to attach. Here's, it's like you can play around with these, and then you can even add a negative transcription factor in there and see what it might do and how that might affect it. Okay. The thing about these is that depending on whatever the environment of your cell is, that's going to affect whether or not these positive or negative transcription factors are going to be used. Okay, so we'll look at an example of those in a couple of minutes here. Okay. So one other view that you have on here that you can do, here we're looking at like protein, it says average protein level, so how much protein is being made in a given amount of time. So starting off here, if we just have a single cell, we're going to have a relatively slow buildup of proteins that are being produced in the cells and in the body. But we can change some things, like we can change how many cells we've got, add a lot of cells. You can see the ones being active are those that are highlighted here, and then our baseline is automatically higher for the number of proteins that are being made. Okay, but this is in a comparison versus time, so while we're making more proteins, the rate is going to be consistent. Now, we could change some stuff, like we could add more positive transcription factor. Okay, and by doing that, that means that more polymerase is going to be attaching to those proteins, so that's going to increase the rate it's going to be made. We could decrease the concentration of the mRNA destroyer, and that should have another positive increase on how much protein is being made. Okay, so you can play around with all these dials. You can decide how strong you want these to attach to the DNA, how long the proteins will survive without breaking down. So it's up to you. I want you to just kind of go around and play with all these. Okay, to get yourself a feel for how the cells work, right? Because we want to see what's the response of the whole body, of the whole organism when transcription and translation is going on. Okay. Okay, so I'm going to give you guys an example here of some gene regulation that goes on. This is going to happen inside of prokaryotic cells, which are bacterial cells. Eukaryotic cells don't do things too much differently, but there are different terms and some different stuff that happens. But the theme is kind of the same with all of these. The idea here is that gene expression, or the production of proteins, transcription, translation, it's dependent on the environment of the cell and maybe even just of the DNA in the nucleus. Okay, so this first example is about a lactose. It's called the lac operon. Lac is talking about lactose, which is a sugar that you find in milk products. Okay, and there's an enzyme that your body produces that's called lactase. Lactase's job is to metabolize or break down the lactose, or the sugar. So typically in bacteria, the gene for this is always turned off. Okay, meaning that the cell does not typically produce lactase. Okay, so how does this gene get turned off? Well, there's a repressor gene that we see here. And the repressor gene, right, to repress means to stop or inhibit something. So this gene here codes for a protein, and that protein can prevent the production of lactase. So How does it do that? Well, it has a very specific shape, and here we see that shape. It looks kind of like something with cat ears on it. This is always active, okay? So this gene is always active. So this gene is on, and it produces this protein, and this protein is always present, and it's always bonded to the DNA right here in this promoter region. And so because this is here, the RNA polymerase can't bind to the DNA and copy the lactase gene. So how do we make lactase then, should be the question. Well, there's another gene that's very nearby here. All these are grouped together. There's another gene that's called this one's going to produce a protein called allolactase, and here we find it right here, and it's going to produce a little protein that's going to bind specifically with the repressor protein. So we've got a repressor gene that makes a repressor protein, and then we've got another gene nearby that will bond with that repressor protein. And look what happened to it. It changed the shape, okay? So remember, form equals function. So we changed the form or the shape of that protein, and so now it has a different function. Before, its function was to fit right here into this spot and prevent polymerase from bonding. But now that it has this new shape, it doesn't fit there anymore, so it can't do its job. Now polymerase is able to bind with the DNA, copy the gene, and now the enzyme called lactase, which breaks down this lactose sugar, can begin being made, and the protein can start breaking down that sugar. So I know it's like kind of crazy, right? But the way this process works, right? It's always off until there's lactose present. So this one starts being made as soon as you drink some milk, right? Or eat some ice cream. This gene starts being made. You change the internal environment inside the cell. It recognizes this sugar, and that sugar stimulates the production of this protein right here, which will deactivate the repressor, and now the gene can start making lactase, which breaks down the sugar. Gosh, I hope that makes sense. It's complicated. That's why I want to try to give this to you right here. This is real examples. So this other example, it's called the trip operon for tryptophan. Okay, tryptophan is it's an amino acid. Okay, so this one worked off of a sugar, and this one's going to work off of an amino acid. In this case, the gene is always on. Okay? In the lactose one, it was always off as a starting position, and in this one, it's going to be always on. Okay? And so in this case, the gene is going to always be producing enzymes for the protein synthesis. Okay? There is a repressor gene also for this, but it's typically inactive. Okay? See the shape of it? It's not going to fit up here. Okay? It's not going to fit. It's all disfigured. So the repressor gene is always off. And the tryptophan gene, the one that's going to make tryptophan, it's always on. So cells are always producing tryptophan enzymes. What's going to change, though, is that once there's enough tryptophan produced by the cell, once the cell realizes that it has enough, then it's going to turn it off, right? The internal environment shifts. So the environment causes a change to how the cell responds, and now it's going to start producing this gene, or it's going to start producing a gene that's going to allow this to change shape and therefore deactivate the gene and turn it off. So it's going to turn it off until the cell needs more tryptophan, at which point it's going to turn off this gene and it can produce more. Right? It's kind of nuts. So think about what I said here on this, okay? think about what I said here on this one. Read through it yourself. Look at the diagram. Study it. Go back here. Think about like how that's going to apply. Try to imagine this situation in relationship to what's happening on these guys here. Okay, so it's going to kind of take some back and forth, but I want you guys to give this a try. Give it some thoughts. Send me some comments about it. Send me questions, and we'll talk about it in class on Monday or Tuesday for sure. But give this a shot and let me know what you can come up with here. All right, so I hope this is helpful. I hope it's not confusing you, but good luck with this, and I'll see you guys back in class on Monday. So thanks. See you later.